Sorry. Okay. So this week uh, we do not have much content to cover. So what, uh, according to the video lectures as well, we'll cover some older concepts, FPA, constraint-based modeling, and related stuff that we have learned for the last 2 weeks. I'll go over them a little bit, and after that, there's one topic for this week, that is the flux analysis of 13C, metabolic flux analysis. So that will go through. So since we have only one topic, I have not kept questions for today. So if you have any questions of your own, you can obviously ask me. Other than that, I'll go through the material, and at the end, I'll talk about that 13C metabolic flux analysis. That's the only topic for today. Other than that, the rest of the part is actually repetition from other week, and we'll basically go through it. Okay, let's get started. So first concept was constraint-based modeling. Just to give, I'll go through this a little bit more faster than last time because this is, a revision session for those topics. But yeah, feel free to tell me if I'm going too fast. So constraint-based modeling, what we learned is we can put some when we have a kinetic model or a metabolic model or any kind of model that we base our assumptions on for a particular pathway or a particular disease or a network, signaling network. So we have a model, and there can be like infinitely many solutions to that model. So we have differential equations in the form of model, and we solve those equations and we find optimal global optimal maxima, optimal minima, or optimal value of the solution that satisfies the whole uh, problem that we are handling. In our case, in this particular case, will be a metabolic network. So we can put various types of constraints on the model. So there can be physiochemical constraints, which includes mass and energy and momentum balance. So we have seen this through various examples, how conservation of mass is used for adding constraints to the model. There can be topobiological models. So for example, steric hindrances. Uh, so crowding of molecules inside the cell. So you cannot have such a high rate of growth of biomass that your cell does not fit all the molecules together inside it. So those kind of constraints are also possible. There can be environmental constraints as well, including pH, temperature, etc. So some reactions, obviously those the enzyme catalyzed reactions inside the cell will be very sensitive to these factors, this pH, this temperature. Everything has to be perfect for the reaction to go through. So environmental constraints can also be added to the model. And the last type of constraints is regulatory constraints. If you recall, we do not use regulatory constraints in FPA, flux balance analysis. We have this we have a different method for integrating regulatory constraints, which is basically what we call as a room, and also there can be RFPA, which we studied in the last class. So regulatory FPA, where you can add these regulatory constraints like which gene activating which transcription factor, and etc. etc. Can add all of that. So that is basically transcription and then translation regulation on and how the enzymatic activity is changing with that regulation. So this is an overview of constraint-based modeling. After that, next comes the point of mathematical representation of constraints. So there can be two types of constraints. There can be balances, which is basically mass balance, energy balance. So basically, the amount of energy or the amount of mass going inside the system should be equal to the mass or the energy coming out of the system, including the one that is utilized inside the system. So basically, laws of mass action. The, the mass flowing inside equal to the mass inside, ultimate like final mass inside plus the mass that is flowing outside. So uh, principles like conservation of energy, conservation of mass, these are like balances. These are like basically uh, the amount that is going in should be equal to the amount that is coming out. Uh, be it in case of energy, be it in case of mass. In mass, we are more concerned about like basically the amount of reactant or the amount of a particular biomass that is going inside the system. Next is the bounds. These are basically numerical ranges of individual variables, such as concentrations of fluxes or kinetic constants. So fluxes we'll talk about later, obviously. Concentrations, we can have certain concentration bounds. So like we know that a particular compound is prevalent within the cell, and if below a certain threshold of that concentration of that compound, the cell is not being able to live. It is a lethal phenotype. So these kind of information that we have from before, we can use these as constraints to uh, bound the solution according to the biological need of the system. So basically, you limit your solution space to a specific range or a specific parameter. So as I said, the conservation of mass is an example of balance constraint. Mass balance is utilized in terms of flux and stoichiometry of each reaction. Stoichiometric matrix is basically a matrix representing all the stoichiometry of the network, of all the reactions in the network. So it is an n by n matrix, where n is the number of metabolites, n is the number of reactions or fluxes, and each of these elements of the matrix is basically the Sijth element will be of reactant I and in reaction J. So basically, your columns will basically your rows uh, uh, will be n cross n, your rows will be the reactions, and your columns will be the no, sorry, I said it ulta. So your rows will be your metabolites, and your columns will be the number of reactions basically. So your m by n stoichiometric matrix will have your Sij element of that stoichiometric matrix will define the ith reactant in the jth reaction. So this we all have studied before. Uh, Next is flux balance analysis. So recall. So we have, this is basically the figure showing is the workflow of flux balance analysis. So we'll have to curate metabolic reactions. You form the stoichiometric matrix. So stoichiometric matrix. Then you define some objective function depending upon what question you are trying to answer, and you try to maximize or minimize that objective function up basically constrained to these conditions that you are putting in. So you have you have all the reactions, you form the matrix, you define your objective function, you apply the constraint, and you try to optimize Z using LPP and try to find the point which is basically depending on your constraint. So depending on your objective function, you can either maximize or minimize the objective function. So for this case, they are trying to maximize it. So this point will be your optimal solution. So first, a metabolic network reconstruction is built. Next, the reconstruction is converted into a mathematical model by forming a matrix, which is the stoichiometric matrix. At steady state, the flux to each reaction is given by the equation S equal to 0. So this is the steady state condition that we follow always. So SV equal to 0. So your V is your vector containing all the reaction rates of all the reactions inside the network. So it's basically your V is in the null space of your stoichiometric matrix. So you're trying to solve for V. An objective function is then defined, which is defined as Z equal to C transpose V, where C is the vector of rates indicating how much reaction contributes to the objective function. In practice, when only one reaction is desired for maximization or minimization, C is a vector of zeros with one position of the reaction, with a one at the position of the reaction of interest. While simulating growth, this objective function will have a one at the position of biomass reaction. And finally, you calculate Z using LPP, and you'll try to maximize or minimize this Z depending on your question and your objective function. So yeah, so this is basically I think yeah yeah. Yes. So next comes the concept of metabolic adjustment, minimization of metabolic adjustment, which is known as MOMA. It is an algorithm for predicting metabolic flux distributions after a gene knockout. So what happens is basically after it assumes that after a gene knockout, the metabolic fluxes will undergo minimal change with respect to the flux concentration of the wild type. Why is that? Because of the inherent property of biological systems being robust. So if you try to delete one of the genes, there is a possibility that certain other genes will take up its function, or so that the this whole system does not collapse because one gene is deleted. So this is a inherent property of biological systems. It is mostly robust. So what the issue this so this on that context, this is a very fair assumption that the metabolic fluxes will undergo minimal changes with respect to the flux concentration of the wild type after a gene is knocked out. Now, what this MoMA does is it employs a quadratic programming to identify the point in flux space which is closest to the wild type point based on the conjecture that the mutants remain initially as close as possible to the wild type optimal in terms of flux value. So basically, this is your green part is showing your FPA of your normal, of your wild type, and this yellow part is showing the FPA of the knockout. And basically, if this is uh, yeah, your basically optimal point, FP of the knockout optimal point, and this is the FP of the wild type of optimal point, depending on the obviously the objective function. What you try to do is you try to minimize these values. So you try to minimize the distance between the uh, your optimal solution for your wild type and your optimal solution for your knockout. So you try you take this is a trial and error method. You try to minimize it through depending on what technical you use. You can even use LPP to minimize this. But yeah, this you can. The whole idea is your knockout should be very similar to your wild type in terms of fluxes. So the differences in the optimal solution should be the lowest. Uh, yeah, I'm not going through the figure in like so the caption in detail, but yeah, last time I went through it, but it's the concept is that you are trying to minimize the differences between your knockout optimal solution and your wild type optimal solution. Uh, to predict a metabolic phenotype, HOMA determines a flux vector in the flux space of phi of a mutant with the smallest Euclidean distance. So in this case, they are minimizing the distance, the Euclidean distance between the two. There can be other distance parameters also that you can take. You can take entropy as a distance parameter. You can take other. There can be many other distance parameters that you can take, but Euclidean is the most common one, and and is also like it's symmetric basically. X to y distance is equal to y to x distance. So this may not be the case for all the metabolic networks that you're using, but this in this case it is feasible, so they've used it. So it's basically just uh, taking the root and summing up of all the residual squares. Basically it is uh, the Euclidean distance between the 2 points. So next is another variation of FPA, which is ROOM, which is basically the full form of regulatory on-off mechanism. It what is interesting about it is it basically integrates regulatory constraints into the prediction of the post-adaptation states. So as we saw, FPA does not do this. FPA does not integrate this regulatory constraints into its model. But there's another variation of FBA to do that. That is the flux variation analysis or flux variance analysis, FVA. But no, no, sorry. There is another way to add regulatory constraints in FVA, which is basically regulatory FVA, RFP, not FVA. FVA is a different thing. You check for uh, uh, various points in the flux space. I'll talk about FVA. Uh, so how is this ROM or room different from MoMA? First is norm selection. MoMA minimizes Euclidean distance between flux distributions of the wild type and your knockout, while room focuses on minimizing the total number of significant flux changes, emphasizing the cell's adaptive cost reduction. It also includes regulatory changes, which is basically the hallmark for this tool. So it includes regulatory changes post in knockouts are minimized by the cell to reduce adaptation costs. This is the assumption that the, that the model runs on. And it operates also under the assumption that regulatory changes can be parsimoniously described by Boolean on-off dynamics, basically and or whatever be the logic. Those kind of gates you can use, Boolean on-off gates you can use, which can be representative of the regulatory mechanism that is going on inside the cell. Uh, uh, the advantage to this is basically it assigns fixed cost to regulatory change irrespective of the material. But in a real system, in a real biological system, this may not always be true. But yeah, this is an assumption that you can make. Uh, it it incorporates implicit regulatory constraints, accounts for regulatory changes by identifying significant flux changes in metabolic reactions. This is essential as efficient expression likely requires adjustment in flux implicating changes in gene expression level, which it takes into account. And the last one is while both MoMA and Room aim to find flux distributions close to the wild type, Room's metric implicitly prefers higher growth rate solutions. This preference leads to more accurate predictions of the post-adaptation states, as evidenced by its ability to minimize significant flux changes while maintaining stoichiometry. So it basically, the whole idea is it will prefer high growth states more than anything else. It will prioritize it over everything else. So uh, it can so if that happens, it is more likely to give a more accurate prediction of the post adaptation states. Okay, so yeah, so this is an example of room. This is the same example that we saw last time. So this is basically this first one is the wild type network that can be obtained by FPN experimental data. This B2, the flux through B2 represents the growth rate. MoMA's prediction for the knocked out network following this basically V6, this network has been knocked out. So this panel, this B second panel B, it's basically MoMA's prediction for the knocked out network following deletion of V6. Rooms, the last one is basically Room's prediction for the knocked out network. Uh, Here also this V6 uh, is basically knocked out here. So the difference is basically room finds changes in flux along only along a short alternative pathway through V5 and V4. So basically here is your V4 and here is your V5. So instead of going from C to E, it is moving from VD to E, preserving the optimal growth of this uh, system. In basically uh, in MoMA's prediction, I think MoMA is predicting. For comparing Room and MoMA's predictions, Room is predicting more growth. It is prioritizing more growth rate in comparison to MoMA. But in general, the basically the major difference in basically MoMA and Room is Room will take into consideration of the regulatory constraints and it will try to maximize the growth rate. MoMA on the other hand, it will not take the regulatory constraints into consideration, and it is not always the case that the maximum growth rate is the ideal solution in case of Room, basically. In all the figures, so in case of MoMA, sorry. In all the figures, metabolic branch point B, so basically this is your branch point, along with the reactions that produce or consume it are highlighted. Note that in Room's prediction, the linearity of the flow is preserved in branch point B uh, as compared to the one as compared to the MoMA's prediction. Uh, Also, MoMA's prediction has a Euclidean distance of 62.8 from room solution and has three altered fluxes with respect to it. And MoMA solution has nine altered fluxes with respect to it. Room solution has an Euclidean distance of 75 from it. So remember MoMA's idea. It is to minimize the Euclidean distance between your mutant optimal solution and your wild type optimal solution. So in case of MoMA solution, it is actually the Euclidean distance is certainly lower compared to Room's solution. So it is 62.8 compared to 75 in Room solution. But also keep in mind, uh, Room solution has only 3 altered flux with respect to A, whereas MoMA solution has 9 altered flux. So that ideas behind this model are slightly different, and you can see that in the results as well. It is very much reflective of what it is trying to prioritize, what it is trying not to what it is not prioritize. So from the results also it is visible that Euclidean distance, OMA is trying to minimize this Euclidean distance, and Room is trying to minimize the number of changes after that perturbation is taking place. In this case, a knockout. So the next concept that we come to is the are the gene protein reactions. We studied this last week only. So basically, there are various kind of associations that can be possible. Direct associations like simple associations like gene to protein to reaction, or there can be like multiple genes forming multiple proteins which together regulate the reaction, or there can be multiple genes forming subunits of a same protein which come and fuse together and which basically catalyze a reaction downstream. Or there can be another association which is basically this is a multifunctional protein. It's one gene giving one protein, but that protein is involved in many reactions. This is a very common phenomenon that we see in cells. This is known as multifunctional protein. This is basically two different genes forming a single protein which is catalyzing a reaction, which is known as multimeric protein, which basically they'll form a monomer of or a dimer of each of dimer of the protein, and they'll fuse together to form the basically each will form the monomer and they'll fuse to form the dimer, or it can be 2 plus 2 4 as well. So there can be many modules, submodules of the protein that is getting formed, but the point is there can be multiple proteins, multiple sources of the subunits and which come together to form a protein which uh, influences a reaction. So yeah, so basically this is a simple association. These are what are known as isozymes, basically uh, uh, two different proteins having a similar function. So these are known as two different enzymes having a similar function. So this is basically isozyme. This is multimeric, and this is multifunctional. So these are different types of gene protein associations, reaction association you can have. So first, we'll talk about two variations versus what is first is metabolic reaction deletions. Second is we'll talk about overexpression. I don't think I have. Yeah. Oh, I've also talked about gene deletions and metabolic deletions. So okay. So FPA, all we know, it's basically uses it can simulate these effects of these in silico gene deletions or knockouts, and the relationship, uh, the bottleneck here that is that most of the genes or most of the proteins are basically either multifunctional or isozymes or multimeric. So there is not a one-to-one mapping between the genes and the proteins and the reactions. So enzyme catalyze reactions, but multiple enzymes can catalyze the same reaction, and also one enzyme can catalyze multiple reactions. And deleting a reaction may require deleting multiple genes, which can alter the metabolic solution space. For instance, removing a specific reaction may improve production rates, but deleting the genes associated with that reaction can lead to a lethal growth phenotype, as other essential reactions may be affected. Additionally, if a reaction lacks known associated genes, genetic engineering may not be feasible as well. So this is basically how they uh, this is basically how they model the framework. I'll come to that later, but there are stark differences between gene deletions and reaction deletions. So uh, this example that is on the left is basically a GPR association for a transketolase reaction, TKT1 and TKT2 in E. coli. These are basically reaction deletions where basically um, you have only in theory you have only stopped that reaction, but the genes are still there. They are functional in the other reactions they were previously doing. So only this reaction is not functional. So this is basically the blue is the wild type solution space, and and TKT1 PTAR reaction deletion. Basically, this is the reaction deletion strain. Uh, this is given in green, and ethanol production coupled to cellular growth is seen at an improved rate in the mutation strain. And also, when corresponding genes, all corresponding genes are deleted to eliminate this particular reaction, then there is no growth predicted from the mutant only, because this, you see, this is the solution space of the mutant. There is no growth. Basically, those were important genes inside the cell which were contributing to different reactions in the cell, and when you knock them out, what you get is a lethal phenotype. So yeah, so it's very much possible that, like, it is very unlikely that when you're trying to delete a pathway or delete uh, a reaction, you delete all the genes that are associated with the reactions, because it is very much possible that the cell will not survive. So yeah, so that was the difference between reaction deletions and gene deletions. Now this is the modeling framework. Uh, so this first panel, this A1, this is a metabolic model, and this is represented by the combination of three model components. So G1 to G5, you see there are 5 genes that are associated with the reaction, and these have basically these are represented by Boolean rules, and with basically you have 5 genes and you have 3 reactions associated with those 5 genes, and you have Boolean connections or Boolean relationships between these two elements. Now, with reactions and the network of reactions define the feasible set of biochemical transformation. So basically, this yeah, the network of reaction defines a feasible set of biochemical transformations. This is basically your solution space, and these are basically your uh, solution spaces for essential biomass precursors. And growth phenotypes are predicted by assessing whether all biomass precursors can be produced by the metabolic network from the set of metabolites from the medium. Gene deletions potentially inactivate reactions, which in turn may reduce the space of producible metabolites. In case where a biomass precursor is no more producible, gene deletion is predicted to be lethal to the medium. So if you compare these two panels, so if you for example delete this gene 5, this last reaction R3 will not go through. So if this doesn't go through, this is an essential it will not produce an essential biomass precursor, which will help in the metabolism of the cell. And if that is not there, the cell will not survive. So this gives rise to a lethal phenotype. So if you delete a gene, there is a high possibility, okay, it will produce a lethal phenotype. So it is it is more like, experimentally speaking, it is advisable to do gene knockdowns than knockouts. Okay, next comes the concept of dead end metabolites. So uh, it is defined as a metabolite that is produced in the known set of metabolic reactions of an organism, and there is no reaction that is consuming it. So it is inside the system, and basically there's no reaction that is consuming, or it is also not that is consumed by the metabolic reactions of an organism and has no reactions producing it. So for example, uh, there are uh, four derived metabolites here: A, B, C, and D. So you see, A is not getting produced inside the system. You do not know the source of it. B is getting produced inside the system, but it is not getting used up anywhere. You do not know where B is going. C and D, you do not know where they are produced, or how they are basically used up. So these are basically dead end examples of dead end metabolites. Uh, Dead-end metabolites are thus isolated compounds within the metabolic network. In some cases, these DEMs can reflect a deficit or an error in how a metabolic database represents knowledge from a scientific literature. And in other cases, the systems level analysis alerts us to areas where more experimental research is required. So basically, if you have more dead-end metabolites, it means that you have very less information about that particular metabolic pathway or particular molecule inside the particular metabolite inside the pathway. So it's basically you do not know from where it is coming and where it is going. So these are what are known as dead-end metabolites. So next comes the concept of block reaction. So what is block reaction? Initial metabolic network reaction of reconstruction often contain inconsistencies due to annotation errors or unknown enzyme functions. These inconsistencies lead to missing reactions in the draft model, creating gaps in the metabolic pathways. Dead-end metabolites, they are classified into two, as I said, root non-producing, root non-produced, and root non-consumed. Basically, this is an RNP, you do not know from where it is coming, and this is an RNC, you do not know where it is going. Yes, that's all. And RNP, RNC's block reactions cause further gaps downstream or or in upstream metabolites. Identifying these inconsistencies, data and metabolites and block reactions is known as the gap finding program. So these are what are known as gap metabolites. Uh, so the if you see this red crosses indicate the absence of some reaction. These dotted and continuous arrows represent blocked and non-blocked reactions. So basically, these dotted arrows are all non-blocked. This solid arrows are basically blocked reactions. Yellow and green circles represent gap and non-gap metabolites respectively. Metabolites are labeled according to its class. So it is basically RNP, this is RNC, etc. So basically, here also the issue is you do not have enough information. So there are gaps in your information, and so those you do not know where the metabolite is coming from or going to, and you have a block. You basically have a uh, dead end metabolite. Now, if a dead end metabolite is present inside your reaction, there's a gap in the reaction, then what you term it is known as blocked reaction. So basically, you do not know how your reaction is going further, upstream or downstream. Okay, next we studied about flux variability analysis. How is it different from FPA? It basically quantifies feasible ranges of reaction fluxes at optimal or suboptimal production rates determined by FPA. It is a generalization of FPA and aids in understanding metabolic network flexibility. It predicts steady-state fluxes of reactions in a metabolic network based on biological imperative, and it always like basically what FPA is. It gives you a range of solutions. It gives you solutions, a space in solution, a particular area in solution space where your all your optimal solutions are there. But FPA quantifies these feasible ranges of this reaction flux as optimal or suboptimal rates, suboptimal production states as determined by the FDA. So, what what are the different steps of FPA? So, first you identify the maximum value of the objective function, and you constrain the objective function to that, and then you minimize and maximize each flux independently inside that solution space to identify flexibility in the fluxes across the alternate optima. So if you have basically n fluxes, you'll have around 2n FPA problems. Yeah, so basically, this is your max. You have a solution space between your max and minimum, and this is your feasible range, and it determines what is possibly based on external measurements and network stoichiometry. And you have an objective function. You minimize or maximize this subject to these. This SB equal to 0 will be your basically steady state approximation. Your VJ is lying between this range, and yeah, basically CT into V will be your Z optimal. And you solve this series of two linear optimizations for each reaction. Okay, here comes the end to the basically the previous topics that we studied, the, the recap of those previous topics. After this, this is the only topic for today's lecture, is basically uh, MFA, metabolic flux analysis, using 13C based methods. So this method is known as metabolic flux analysis using stable isotope tracing. So it is a powerful tool for quantifying metabolic flux analysis. Is a powerful tool for quantifying intracellular metabolic pathways in microorganisms. Based on stable isotopes tracing, MFA allows the measurement of metabolic fluxes in central carbon metabolism. So the idea is to trace. The idea is you label one of the carbons of a particular molecule, say glucose, and you want to trace how is this glucose involved in different reactions and what is the end product of this. Basically, how much of the original labeled isotope that I get in the end. This is the in general overview. So, what are the principles associated with this 13C based flux analysis? So, first is isotopically labeled substrates such as 13C glucose are fed to growing microorganisms. So, you grow them in a petri dish in agar or anything, and what you do is instead of giving inside the agar that there's glucose obviously. So, instead of having glucose, you have a radio labeled isotope of glucose, which is basically 13C. Then what will happen is alternative metabolic pathways will produce characteristic 13C patterns in common intermediates. And then what you can do is you can use mass spectrometry to trace the stable isotopes in protein bound amino acids over extended cultivation periods. So at different time points you can take out and see. Basically you do mass spectrometry and see how the stable isotopes are propagating. There are two methods to quantify these fluxes. So first is the direct calculation of flux ratios using mass isotope patterns, and second is the indirect estimation of network-wide intercellular fluxes from physiological data and mass isotope patterns using computational models. So we'll see about how they do it. So first is the experimental design. So cultures are grown in 13C labeled glucose to achieve steady state physiology for several generations. So instead of glucose, they give them 13C glucose over multiple generations so as to achieve this steady state physiology. Batch or continuous cultivations are employed based on growth requirements, and measurement of substrate uptake and product formation is determined from concentration profiles inside the culture supernatant. These steps provide crucial physiological data for flux calculation. So basically, what they do is they take the microorganisms, they basically what do you say centrifuge. They centrifuge it inside a centrifuge, and after that they collect the supernatant of the basically your centrifuge product. You collect the supernatant and you check how much difference in concentration is there for glucose and whatever marker you're using to check for the differences in concentration. You'll get an idea about the rate of substrate uptake and the product formation. Also, like if there is some product formation, you'll get that in the supernatant as well. So once the entire of the biomass is synthesized from 13C glucose, then you have to wait for generations to have 13C glucose to like basically in every part of the biomass of the cell. The cell pellets are collected and they are hydrolyzed, and what they yield are large quantities of protein derived amino acids. And these amino acids are chemically derivatized with what is known as TBDMS. It, it's a very hard name, third butyl dimethyl silide. And what it does is it makes it more volatile, and this can be if it is more volatile, then it is easier for in gas chromatography techniques to basically separate these because these are volatile. And after that, you do mass spectrometry analysis where these derivatized amino acids are subjected to electron impact ionization at the interface between this gas chromatography chamber and your MS chamber. And basically, this fragmentation of amino acids will occur here, resulting in approximately 5 fragments with overlapping carbon backbones. And then you do mass spectrometry and you can extract these 13C patterns independently of all the fragments. After that, your final data set will include information from about 60 to 70 amino acid fragments, and it will involve quantification and interpretation of how 13C patterns are present and how you can use it to describe different, use it to determine different metabolic fluxes. This is basically the whole experimental design. Before we move on to the results, I just want to have one slide which clarifies the meaning of these. So there are two uh, concepts here. One is isotopomer and isotopolog. So according to IUPAC, there are two definitions. These are the definitions for this term. So isotopolog is a molecular entity that differs only in isotopic composition, the number of isotopic substitutions. So for example, CH4, CH3D, so one H is basically replaced by D, which is heavy hydrogen, which is deuterium. So CH4, CH3D, CH2D2, these are all isotopologues. Basically, one of the isotopes have been replaced by another isotope. In case of isotopomers, these are isomers having the same number of each isotopic atom but differing in their position. The term is a contraction of isotopic to isomer, which is the, where isotopomer comes from, and it can be either constitutional. Examples, say for example, CH2 DCH double bond O and CH3 CD double bond O. So here the difference is, here we have a methyl group here. We do not have a methyl group here. But instead of that, we have the extra hydrogen is with this carbon. But here, this extra hydrogen is basically included in basically this ethyl group. One extra hydrogen is included with makes it a methyl. It can be stereoisomers as well. This R and S forms this basically cis transforms as well. There can be different types of these isotopomers, but the general idea is in isotopolog, it's basically one isotope is replaced by the other. In isotopomer, the total number of isotopes should be like of a particular atom should be equal, but they can differ in their position. So the total number of isotopes are equal. There is only one D. There is also only one D. But instead of an ethyl group here, we have a methyl group. That's the only difference here. So isotopomers will have the same molecular mass, but isotopologues won't, because hydrogen and deuterium will not have the same molecular mass. And in a mass spectrum, isotopologues will form separate peaks because they will have separate molecular mass, and each of these peaks may contain several isotopomers. So that is possible. Um, just a second, I'll be back in 2 minutes. I just have to visit the washroom. Just give me a minute, I'll be back. Sorry for that. So we saw what is the difference between isotopy and isotopy. Next is basically what is a mathematical and statistical basis for this flux interpretation. So there are mainly two methods to do this. One is global iterative fitting, and second is local flux ratio analysis. So what are the steps involved in global iterative fitting? So first, a system of balance equations is constructed to describe the propagation of 13C in metabolism, considering each isotopomer specific to specific 13C and 12C patterns. Thousands of these topomer balances are formulated for metabolites with n carbon atoms, and the equations are partly nonlinear due to biomolecular reactions and then numerically resolved for fluxes through iterative fitting. What is iterative fitting? Starting from measured extracellular rates, flux cases are used to simulate these 13C labeling patterns, which are compared to the measured data. So you have basically your your extracellular rates measured from the centrifugation, and you can measure how much uptake of the substrate or product formation has been there. So you have that idea, and then you can make some flux cases which you can use to simulate your 13C labeling patterns that you have got, which are compared to the measured data. This is basically a trial and error process and an iterative process, and it continues until a satisfactory match is achieved, resulting in a comprehensive flux map with model estimated absolute reaction rates. Simplifications in modeling, the details of isotope patterns help in reduction of computational demand, but may lead to incorrect flux solution. So it is possible that do not you do not analyze every isotope pattern, isotope of the particular, in this case, carbon that you have. So you do not say you only look at 13C, do not 12C. So that can lead to incorrect flux solution. So it'll reduce your computational power, but it is more advisable to have an idea about how all your like labeled and your unlabeled isotope flux is changed. Detailed isotope balancing as a general example of iterative flux treating is employed using software like 13C Flux. So in the next example that I'm going to show you, they have used this 13C Flux only, sorry, for global iterative fitting. Next is basically this is another type of uh, fit, basically your flux estimation is using local flux ratio analysis. So it is a complementary approach to quantify ratios of flux to converging pathways independent from the extracellular rate measurements. Algebraic equations are developed to relate relative flux amounts at pivotal nodes to 13C labeling patterns in product of converging pathways. Ideally, one flux ratio equation is desired for each of the alternative pathways in the network. It is less susceptible to ill-defined network models due to local nature, because it is like basically alternative ratio equation is for the alternative pathways in the network. So it is local in much more localized in nature, and it is less susceptible to ill-defined network models. And historically, equations were manually derived, limiting availability to model organisms and special conditions. And you can use software packages like Flux. This also they have used, and which can be used to calculate flux ratios from 13C labeling patterns, especially for model organisms on specific substrates. For this case, I think it is only glucose. And recent advancements aim to automate the retrieval of flux ratio equations, easing the development stage for new organisms. Now, this is an overall workflow for this 13C metabolic flux analysis experiment. So you basically have a batch with basically in one part you will have your wild types, and then you do some 13C labeling experiment, and then you harvest, then you hydrolyze the proteins, direct amino acids, and you do GCMS, basically gas chromatography, mass spectrometry of the amino acids, and you check for these 13C labeling patterns. In flux ratio analysis, it's basically you measure the flux ratios from which part of the pathway is the highest ratio of the flux coming, or you do isotope balancing and iterative fitting, and you do a trial and error method, and you find the fluxes through each of the pathways. Flux ratio analysis delivers relative contribution of the converging pathways. So if a pathway is like this, two pathways are converging in a particular product which is getting utilized in further downstream. So flux ratio analysis can give you the relative weights of what the how much percentage of flux is coming from that particular pathway and how much is coming from the other pathway, coming adding up to be 100%. Um, an iterative fitting provides absolute rates of fluxes in metabolic pathways. In, for example, this is one unit, and this is a glucose pathway that is shown. This EMP is basically Emden Meyerhoff Parnas pathway, and this PP is basically pentose phosphate pathway. So in a glucose, there can be I'll show you in the next slide. So basically, how do you calculate your flux? Before going to that, I just want to show you the network. So this is glycolysis. So glucose to glucose 6 phosphate, fructose 6 phosphate, then triose, glycerate, PEP, and ultimately pyruvate. Pyruvate is basically also formed from gluconate. I'll come to that later. This is, on this side is basically the pentose phosphate pathway. There are five basically pentose phosphate pathway, which is connected to glycolysis to pyruvate, and also it is obviously from gluconate you can form glucose, but this is not a very reversible reaction. But gluconate can very easily form pyruvate, so it is connected here. This is basically the Krebs cycle. Acetyl-CoA is your starting point, which goes on in a cyclic manner, and basically you form ATPs. So this is the whole network that they are concerned about. So there are some ATP balances also. You need to check from where the ATP is coming and going. There is NADPH also. You have to check, like these are basically phosphatases and kinases and phosphatases. So either they add a phosphate group to the molecule or they remove a phosphate group from the molecule. Can be either. So these need to be balanced as well. And so this is the whole network that they're looking at. So when coming to flux calculations, uh, this is basically is, in the paper they describe this pathway in detail. What they did was just extra something they added was they added the ED pathway and also this glycosylate shunt. Basically, shunt is a resistant you can use to basically short between 2 points. So this is what it is exactly doing. So between isocitrate and malate, glycosylate is acting as a shunt. Then you don't have to go to from isocitrate to alpha ketoglutarate to succinate to malate, then again to oxaloacetate. You can directly go from isocitrate to glycosylate to malate. So glycosylate is acting as a shunt here. Okay. Uh, from this network, a stoichiometric matrix containing 26 unknown fluxes and 23 metabolite balances, including 5 balances for metabolite measured substrates and product biomasses, glucose, acetate, CO2, O2, and other and the cofactors of NADP and NADPH. So 5 constraints, balance constraints they have put: biomass, glucose, CO2 and O2, and also for these NADPH and NADH. Metabolic fluxes were used as additional constraints for solving the matrix on the basis of the extracellular rates. Sorry, the rates is missing. And this is how they solved basically the flux ratios. So the following modifications were necessary to derive the flux ratios from mixture of this 13C glucose and basically unlabeled 13C and one 13C glucose. The upper bound of a fraction of pyruvate originating from mallet was determined as basically pyruvate between reaction 2 and sorry pyruvate minus PEP by oxaloacetate minus PEP. So basically your PEP is here, your pyruvate is here. So pyruvate minus PEP and your oxaloacetate minus PEP, because you see oxaloacetate has a direct reversible reaction with PEP. So the upper bound of the fraction of pyruvate originating from mallet, so that was given by this expression, pyruvate minus PEP by oxaloacetate minus PEP. The numbers in the parenthesis denote the number of carbon positions. The fraction of PEP molecules generated through glycolysis was assumed to have an intact carbon backbone, and 50% of the molecules are labeled at position 3. Alternative pathways that generate PEP produce either intact and unlabeled PEP molecules, that is from the ED pathway, or cleaved molecules, that is from the transketolase, transaldolase, or PEP carboxylase. So these information is very important to basically infer your results. So you need to know this, that the fraction of PEP molecules generated through glycolysis, basically 50% of the molecules should be labeled at position 3. And if it is generated through alternative pathways that generate PEP, they produce either intact PEP or unlabeled PEP molecules or cleaved molecules. So if you know that the PEP that you are getting is coming from glycolysis, then 50% of the molecule should be labeled at positions, because you can work this out easily. Like if you do a little bit of biochemistry, like how these reactions are going, you can easily trace the radial labeled carbon. So depending on the product or the biomass you are getting, you can use it to basically predict from where actually the source of the carbon is. So either it is coming from the glycolysis pathway, or it is coming from this ED pathway. So if it is coming from this ED pathway, it will either be intact or unlabeled PEP molecules or cleaved molecules. But if it is coming through glycolysis, then 50% of the molecules will be labeled in position 3. Okay, so this is how the flux calculations are made. So there were many constraints, obviously. Total 8 equations were added, which were added as constraint. So first is the fraction of serine originating through glycolysis. Second is the fraction of pyruvate originating through the ED pathway. The fraction of OAA. OAA is oxaloacetic acid from PEP. So basically this connection, oxaloacetic acid and PEP. And the fraction from PEP from OA, because this is a reversible reaction. So basically, you can go from OA to PEP or PEP to OA, depending on your need. And this is a reversible reaction, so both should have bounds. The upper and lower bound for pyruvates originating from malate, and upper bound from oxaloacetate originating from glycosylate, and upper bound of PEP originating through the pentose phosphate pathway. So these total 8 uh, constraints were put on the model, specifically these 8 equations which are described here. And to calculate the rate vector containing the metabolic fluxes, the mass balances for the metabolites were constructed according to this equation: NV minus B equal to Rm, where basically N is the stoichiometric matrix, B is the vector of the measured fluxes, and your Rmb is the vector of the residuals of the mass balances. And if you take this equation NV minus B equal to RMB and you substitute here, so it's basically you basically modify all the equations, and what you get is something like this set of 8 equations again. And then what they did was they basically used the sum of the weighted square residuals of constants from both metabolite balances and flux ratios, and they minimized using MATLAB functions. So this is how the results look like. So basically, you see each of the pathway they have calculated fluxes for each of the pathways, and different conditions under different conditions also they have calculated fluxes. So metabolic net fluxes are estimated with the 13C constraint of flux balancing and comprehensive isotope balancing. These are the like this is hmm, yeah this is basically 20% having 13C, 80% unlabeled. This is 100% having 13C, and then you combine constraint from the first two experiments, and then you use 2080 20% 13C and 80% of an isotope of 13C. Uh, yeah, metabolic net flux is estimated with 13C constraint flux balancing values and comprehensive isotope balancing from the same 2080 experiment of exponentially growing E. coli in a bioreactor culture. Bold face numbers indicate fluxes of metabolic precursors to biosynthesis. So see this alpha ketoglutarate, acetyl CoA, pyruvate, PEP, these all fructose 6 phosphate, triose 3 phosphate, even the pentose 5 phosphate. These are essential like upstream molecules which basically produce biomass precursors or sorry metabolic precursors, so which are essential for the cell. So these are indicated here, so you can get that information as well, like if you have your basically hmm, yeah, basically from where your flux is coming, and also if that flux is involved in a metabolic precursor synthesis. So yeah, that's all for today that we have. So we went through the some older concepts, and we looked at how they calculated how the this paper actually is given in the in SIR's uh, drive as well. So I'll just show you it here. Yeah. So this is the whole protocol that they have followed. This figure is from here. So you can take a look at this paper. So this is the 13C flux that I was talking about. Uh, it's a set of tools for isotope balancing and global iterative fitting. You can take a look at this. Basically, yeah, this is another concept that I talked about, this fiat flux. Uh, from raw GCMS data, you can basically calculate flux ratios in central carbon metabolism depending on organisms and or isotopic tracer. So this is also a useful function or software in MATLAB. And yeah, it contains all the uh, like details required for this study. And also there is another paper, I think this one. Yeah, this one. So this is also this figure is all actually taken from here. So yeah, so they here they have used some software called this Metaphor, but it is basically a flux ratio analysis tool. So don't get confused by that. Uh, Here is the pathway that they have studied, and here are the formulas that they've used. Those equations are from here only. So you can take a look at this paper as well. This is also a nice paper. This is what is the name? Yeah, this is from Fisher and Zamboni. Zamboni alone has another uh, C13 MS paper, but this paper is quite good. You can, if you want more details on how this flux analysis and calculations are done, you can take a look at these papers. But yeah, other than that, I think this is all for today that we have. We do not have any assignment questions or anything because covered very little topic in this week. But yeah. Do you have any questions?
So yeah, just this thing. I mean, for the global iterative setting essentially, so the prerequisite is this well-defined network, right?
Yeah.
So we can only do that once we have this like properly defined network, like only on that. Like otherwise, it's basically this flux ratio analysis.
Basically, yeah, yeah. You need that complete network because uh, it what you are doing is basically you have a particular uh, what to say from that from the cellular medium you have identified
Yeah,
how much substrate is getting uptaken or how much product is getting formed. What you are doing is you have the whole network, you have an idea about from where it is coming and everything. You have certain flux assumptions of the network, and you do kind of a trial and error. You fit it, you see how it is fitting. If not, you do another set of values, you try to minimize errors. Whatever is the point, it's basically a card fitting problem. So it's a trial and error method. But yeah, you need to have the whole network to know like what to have an idea about these estimates of fluxes that can pass through each reaction. So you if you do not have that, you do not have that starting point to basically fit the data and do this iterative. You need to have an idea about how what kind of fluxes are going in. So that you can use it as a trial data and
yeah,
fit.
okay, yeah,
Okay. Okay
yeah,
then. Yeah. So I decided against taking the extra class that I talked about because
okay, okay,
it is clashing with some of my schedules. But yeah, if you have any things that you want me to cover, because in the last class also it will be like this only because we'll have very little uh,
yeah,
to cover. So if you have anything that you want me to go through again, I can go through in the last class. You can let me know in the next or the next class. This is I think the 10th. So you can either let me know in the next class or you can mail it to me if you have any topics you want
yeah.
me to cover.
So I'll do one thing. Like I'll just compile. If I have any doubts, I'll just compile it and then send it to you before this weekend, so that if
Yeah, yeah,
possible,
that will be
maybe
good.
next
That
week, nah, next class it will be easier for you.
not next class. I think next class will be the 11th one. I'll do it in the last
Yeah, next
class,
one. Yeah, yeah, 12th class. Yeah. So
12th.
I'll mail it sometime before
Yeah,
either the
yeah, yeah,
next class or
yeah, yeah.
next weekend or something like that.
Yeah, I'll need actually like 4 days, 5 days
Yeah,
prior
yeah,
should be enough.
yeah, yeah. Sure. Yeah. Thank you.
Okay, then. Thank you for joining.
Yeah,
See you
yeah.
next week.
See you.